we've got these 2 bird programs, one with the heath hen, one with the pigeon. We've had people interested in maybe moving on to the great auk. And the more diversity of animals we can start working with, not only enables more de-extinction, but enables more ability to just save species we have before they get into problems of becoming extremely rare, as well as saving species that just can't cope with climate change or disease or invasive species, or even actually being able to use these technologies to humanely control invasive species. You mentioned, gene drives and whatnot. Those are some of the solutions people are coming up with to try and combat pest species.
Say, before we go into gene drives, just so people don't get lost.
We probably shouldn't go into gene drives. That needs its own phone call.
So let's not go in that direction. What I wanted to ask though, just quickly, is this technology is quite different to the cloning of Dolly, but can it also be applied to say fish and lizards? Maybe insects?
So people do use the same germline transmission technique that we've talked about for birds in fish. It's actually being explored right now for the conservation of sturgeons, which are critically endangered everywhere.
We still eat them.
Yeah. And it's likely the technology that would work for reptiles. No one's really done this with reptiles. Cloning works for amphibians to a degree. There's some challenges to overcome there. But people have done germline transplants in amphibians as well. So this germline transplantation actually would work for a variety of things. And actually just last year, in 2019, a team of scientists published that they had accomplished germline transmission in goats, cattle, sheep, and mice with a new technique that they used. To date, until they had done that, the only mammal that anyone had ever succeeded with that was mice, like 40 years ago. And then no one got it with anything else. And they've been able to do it with males. I should say that they've done it with males right now only. They have not yet done it with females. But, that's still, let's say you have a wild species of goat that's facing a challenging disease, right? Well, what they did was knock out a particular gene that makes the germ cells in a regular goat so that it no longer has any of its own. So you can take the cells from a different goat, put it in the male, right? Mm-hmm. So let's say you work with the germ cells in a petri dish, you edit in an antibody that allows them to overcome that disease. You then, for the wild goat, you then put the edited wild goat cells into one of these special surrogate domestic goats, and it produces a bunch of semen. Mm-hmm. You then use that to artificially inseminate a whole bunch of wild females of that species. And all of the offspring now have inherited one copy of this new antibody. And you start spreading a trait into a population. So, there's tons of ways in which this same technology could start helping species around the globe. But we definitely need to continue the innovation, work with more. Something we're doing with, and this comes to some work that Revive and Restore actually just released last August, was with partnership with a company called Viagen Pets and Equine and San Diego Zoo Global. We cloned the world's first endangered Przewalski's horse. And that's actually the very first time a species has been cloned for the purpose of genetic rescue in history. And the horse's name is Kurt. And he is alive and thriving today. I'm actually going to go visit him this month.
That's something I wanted to ask you about, actually. So Dolly, who was the first clone, died quite young. Was that
Dolly was not the first clone.
Okay. Okay. You have to tell me about that. But
getting
also...
this year I've done research into 2 big topics. So one is cloning and the other is translocations and whatnot. And that's how I was able to tell you stuff about bison have been returned to Spain after 15,000 years. But you're hitting the questions that have been all about my work this year. So first I'll say this is that one, we did produce the clone Przewalski's horse, and the amazing thing about that horse is that we used cells that were frozen 40 years ago. Mm-hmm. So that is a species that became extinct in the wild every zoos worked very hard to repopulate those and reintroduce them in Mongolia and China and Russia and Hungary. They have wild, free-roaming herds now. It's not out of the woods yet, but it is recovering, right? It was extinct in the wild. It was downlisted to critically endangered. It's been recently downlisted to endangered. I mean, it's going, it's going in the right direction. But all 2,000 living Przewalski's horses, whether they're in zoos or the wild, are descended from 12 original Przewalski's horses captured in the early 1900s. And it's a little more complex than just that, but to simplify it, the breeding in the first few decades managed, to really make just a lot of loss of genetic diversity for that species. And so we reached back into the past to grab the cell line of a male called Kuporovich because it was known that he had unique ancestry to 2 of those 12 original horses.
Where did that DNA come from? Sorry,
those were cells preserved at the San Diego Zoo.
Okay,
and Kuporovich was bred. He does have some living descendants, but he was not bred very much. And so he has this unique ancestry to the original founders that is just not represented very much in the population anymore. And so that was the reason we decided to choose him, to be able to actually get a second chance and get his diversity spread throughout the population better. Because, in the 4 generations since he died, since he was born and was breeding, every generation only inherits half of his diversity, right? So, I mean, it's like this issue of genetic drift in small populations that we face. So we cloned him, and what's amazing to me is, so we named the clone Kurt after the founder of the frozen zoo at San Diego, Kurt Benerschke. And Kurt is thriving, he's doing really well. When he grows up and breeds, it'll be the first time a clone has ever been resurrected from the past to bring back diversity. And the neat thing is he was born the same year as his great-great-grandchildren. The technology to clone Kurt was basically very similar to the technology to clone Dolly. And you hit on one of the key things right away is there's a lot of misconceptions about cloning as a technology. Mm-hmm. One is that Dolly was the first clone. Dolly was not the first clone. Dolly is the most famous clone because of the breakthrough that she represented. So the first cloned animals ever produced were actually cloned in 1957. And they were frogs.
Wait, when was Dolly?
1996.
I mean, that's they won by quite a bit.
Yeah. And people were cloning different types of frogs and salamanders and fish in the 60s and 70s. And then in 1981, a team of people successfully cloned mice. And those were the first cloned mammal. And then they cloned sheep and cattle in the late 1980s. So actually the first cloned sheep were cloned in 1986. The thing that made Dolly different was that all of those clones for nearly 40 years had been cloned using embryonic cells, which embryonic cells, if you know anything about, embryos and stem cells and whatnot, they can become any tissue. They can, they're very plastic. Whereas if I take a cell from an adult sheep that is a skin cell, that cell is programmed to be skin. It is not programmed to be muscle tissue or fat tissue or brain tissue, right? I mean, so it's like the challenge was, how do you get an adult cell to be reprogrammed to an embryonic state? And Dolly was the first organism ever cloned from adult cells. And she did die young. However, she did not die because she was a clone. Not a lot of people know that Dolly, she lived to be about 6 years old. She produced offspring. She was fertile. She reproduced. She had babies. Her babies were healthy. She was largely healthy, but she was confined to living indoors, which gave her arthritis, and also what made it, at a greater chance of risk of being exposed to disease if disease came through with certain animals. And that's what happened, is that there was an epidemic, just like COVID, going through kind of sheep farm industry and whatnot. And Dolly they weren't able to move her to a safe place before she was exposed. And she got exposed to a virus. The viral infection caused her to grow tumors in her lungs. And so it was that killed her. And what not very many people know.
Damage that's done to your sort of research. I mean,
oh God, it's the social perception of cloning because of the misconceptions of what happened with Dolly, and then other things since, right? So like in 2001, the world's first extinct subspecies was cloned, but it died 7 minutes later. The first endangered species was cloned in 2000.
This was the Ibex.
That was the Ibex, yeah. The first endangered species, a type of wild cow called a gaur, was cloned in 2000. It died 2 days later. I mean, these because they were dealing with endangered species, these, once again, like Dolly, they got a lot of press. They got a lot of attention. And the gaur died of disease. The Ibex did die because of an abnormality, which happens with clones. But the thing that people don't appreciate about those projects is those were not projects to the scale of what people working with sheep or cattle or cats or dogs do. They were working with low funds. They couldn't work with as many animals as they could have. And absolutely, the team that cloned the Ibex, if they had had more money and more resources, would have produced healthy babies.
And what's the status there? Has that continued since... That was 2003?
That was 2003, yeah. They renewed some experiments in 2013. Once again, did not have enough resources to really do a large-scale experiment. Didn't get any pregnancies there. And so far, that project has kind of been just shelved because a related species of Ibex has actually been introduced into the former range of the extinct subspecies. And they're thriving. They're doing really well. So there really might not be a great need to kind of bring that extinct species back because its relative is doing its job in the environment. But the truth is that in the last 20 years, people have cloned and produced healthy, fertile coyotes, healthy wolves. They've cloned a European mouflon that, once again, sadly died of...
Sorry?
It's a wild type of sheep in Europe.
Okay.
And that clone sadly died young as well, but it did survive to adulthood. It got hit by the same disease that killed a lot of sheep in that species. There's been a lot of good successes with cloning, but, because of some of these misconceptions and these how people think about how cloning has been done, it's really prevented it from ever really being used in conservation, because a lot of people working in the conservation field, scientists especially, they're not even all aware of how much work has been done in cloning and how it can be used. And so, it's been 24 years since Dolly was cloned. And just now we've finally, done genetic rescue cloning for the first time. Mm-hmm. And so Revive and Restore, we're really hoping that we can, kind of work towards a new era where people really start using these technologies and adopting them more in strategic ways. And we've been kind of developing multiple programs to kind of do this. And I think the biggest thing about the misconceptions of Dolly that not a lot of people know is that the same cells that were used to clone Dolly were actually used again several years later, actually more than a decade later, to make more sheep clones. And so Dolly has 4 living identical sisters, and they have all lived to old age.
I see.
But they were raised in a pasture, outdoors. Mm-hmm. Only one of the 4 clones has developed arthritis like Dolly. Mm-hmm. And it shows that the arthritis is not from the cloning process. It's not genetic. Otherwise, they all would have got it. Mm-hmm. Right? I mean, so it's really about environment. And so, not a lot of people know that whole story about what's happened since Dolly.
I certainly was not aware of it.
Cloning also, I should say, is it is just a tool, right? So it's a reproductive tool that if you have living cells of something, you can use it. If you have surrogate mothers and things that you can put the clones into, you can use it. A lot of resources, things have to happen, right? So nobody's going to be cloning woolly mammoths or cloning extinct species because those all died out long before we were able to preserve their cells in an ideal state. But by preserving as much tissue samples as we can of endangered species today, we can use technologies like this germline transfer or cloning to be able to, in the future, reach back into the past, grab some diverse individuals and help those species.
So is that a program that's underway? Are there groups now that are there genetic libraries that are being put into place?
Yeah, they started in the 1970s, actually, and they're all over the world now. So one of the most famous is the San Diego Zoo's Frozen Zoo, but there are biobanks of tissue and cell cultures all over the world. There's, I think there's the Frozen Ark, which is a British-based biobanking system of multiple institutions. There's the Global Genomics biobank, which has its epicenter at the Smithsonian. There's a number. There's thousands of tissue samples of species, of many different species that have been preserved, which with the right reproductive technologies are all potential resources to aid the recovery of those species if they ever become endangered in the future, or for the endangered species, start possibly helping them now in different ways.
Escaped Sapiens.